Botanical Society of America's Emerging Leaders Lecture, and as you know, Dr. Ben Blackman was last year's winner of the Emerging Leaders Award, and this year he'll be giving a lecture. Ben is currently an assistant professor at the University of California, Berkeley. He did his undergraduate degree at Stanford University. I believe he graduated in 2001 and then spent a couple of years as a technician in Dave Kingsley's lab, who works on the evolution of sticklebacks, particularly the evolution and development of sticklebacks, which I think shaped a lot of Ben's thinking. I first met Ben in the summer of 2003 at a new phytologist meeting on plant speciation, and Ben and I went on a collecting trip where we collected the great white stickleback in Nova Scotia. But somehow Ben decided to move away from stickleback and come to Indiana University for a PhD, where he worked with me and with Scott Michaels, who is a molecular biologist working on flowering time. And so Ben did some wonderful sort of cross-interdisciplinary research on the evolutionary genetics of flowering time variation in wild and domesticated sunflowers. He then did an NSF-funded postdoc with John Willis at Duke University and Dan Roxar was a joint postdoc with Dan Roxar at Berkeley, where he moved over to work on Mimulus, and he went from there to University of Virginia for a couple of years and then moved to Berkeley in 2016. Ben has been a highly productive researcher. He has more than 30 publications. He also, and I think this is the most remarkable thing, is that he's managed to get lots of money from NSF, which hardly seems fair in this current funding climate. I'm in Canada, so it's okay. But he's robbed all of you. So, as you'll see from Ben's talk, he takes a highly integrative approach to understanding how plant development responds to seasonal and diurnal environmental fluctuations and how these plant responses evolve during adaptation or domestication. As I've already mentioned, he works on two sort of classic systems, a diverse system, sunflower and monkey flowers, and he's produced many novel insights spanning from the nucleotide to the population level in the evolution of photoperiodic flowering as populations adapt to local habitats, the contribution of gene duplication to evolutionary innovation during domestication, and the biology of soil tracking. I also want to I think it's really important to also emphasize Ben's contributions to teaching, outreach, and professional service. Since defending his PhD, Ben has mentored over 70 undergraduate students and high school students. Many of these diverse students have continued on in STEM fields, and Ben was recognized as an outstanding faculty mentor by the Louis Stokes Alliance for Minority Participation in STEM in 2015. He has organized several regional meetings and symposia and served on the BSA Public Policy Committee since 2013. Among his achievements in outreach, Ben mentored K-12 planting science teams for 5 years, maybe more now, and implemented a discovery-based genomics curriculum for high school interns at the National Museum of Natural History. He is now the lead organizer of Walking with Wildflowers, a new citizen science initiative for monitoring plant phenology along the Pacific Crest Trail. So please join me in welcoming Ben.
Well, thank you, Lauren, and thank you all for coming. Can you all hear me in the back if I step away from the microphone? Excellent. All right. It's really wonderful to have my lab's research be recognized in this way by BSA, and I'm really excited to tell you about some of the questions and projects we've been pursuing in the lab over the last few years. And I'm going to start by addressing what I mean in my title here by the predictable environment. Often we think about the environment as entirely unpredictable. Swaths of forest can be decimated by new pest invasions or new pathogen strains in a single season or two. Or an individual tree has no way to anticipate when, wham, it's going to be uprooted by a tornado and flopped to the ground. But in many ways, environmental change is predictable, and that's because that change is predictable fluctuation. So on a daily basis, every 24 hours, we know that the sun will set and you can bet your bottom dollar that the sun will come up tomorrow. And with that, there'll be changes in the availability of light, warmth, moisture, as well as associated changes in predators, pollinators, herbivores, and other aspects of the biotic community. Likewise, on a seasonal time scale, it's the height of summer here right now, but as I'm sure any native Minnesotan will tell you, winter is definitely coming. And with that, there'll be shorter, colder days, differences in the amount of type of precipitation, and associated changes in community interactions. And then finally, even on even longer time scales that can span generations, there are cycles in the environment, like El Nino and La Nina, that lead to predictability. So here, warmer than average years tend to be followed by warmer than average years, and colder than average years tend to be followed by colder than average years. We can think about similar cycles in community interactions, for instance, with lack of Volterra dynamics and predator-prey interactions. And these fluctuations pose a challenge for organisms to cope with, but they also provide an opportunity in that they provide reliable information that organisms can respond to with phenotypic plasticity, and organisms often evolve to do so. So on a daily basis, there are many developmental and physiological rhythms that show daily rhythms. I'm sure you're all familiar with your own daily rhythms, and if there's any truth to the slide from the UNC School of Pharmacy that I just pulled off the web, we're now in a period of high alertness, which is good for me because you're all paying attention. We're a little bit far away from the area of best coordination, so please forgive me if I fall on my face. Plants have daily rhythms in their activities as well, both in terms of growth, how they interact with pollinators and with herbivores. And an example of a dramatic daily rhythm that we study in my lab is solar tracking, or diheliotropism, in the common sunflower, Helianthus annuus. Sunflowers face east in the morning. Over the course of the day, the apex follows the relative position of the sun to face west by dusk, and at night they reorient back to face east in anticipation of dawn. Okay. So on a seasonal level, organisms also respond to the progress of the seasons, especially in terms of their life history, whether it's when they germinate, when they flower, or when they set seed. These are often times that they occur at times of year most favorable for growth and reproduction. And on multi-year time scale, we also see responses across generations that are known as transgenerational plasticity, where an individual genotype can produce different phenotypes depending not on the environment it's experiencing, but the environment experienced by its parent. For instance, in many species, the offspring of parents that have been subject to herbivory the offspring develop better defended both morphologically and chemically, even in the absence of herbivory. And we're beginning to study this phenomenon in my lab with transgenerational plastic responses to climate. And this is a project started by Jack Colicchio, a postdoc in the lab, and Ian Anderson, an undergrad in the lab, in the lobe-leaf monkey flower, Erythranthe laciniata. And what they have shown so far is that when offspring are grown in cold environments, they grow more quickly when their parents were also grown in cold environments. And likewise, when the offspring are grown in hot environments, they grow more quickly than when their parents were grown in hot environments. To see more of these responses for different phenotypes and understand how we want to get the genetics of this, please go see Ian's talk on Wednesday afternoon. The last thing I want to point out in this slide, because this is the last thing I'll talk about for transgenerational plasticity today, is just the point here that in the hot environment, these three different genotypes in different colors have different magnitudes of their transgenerational response. And this brings me to my next point, which is that responses to predictable environments evolve. Okay? The predictable relationship between fluctuating environmental conditions and the time of day or year that are favorable for growth, development, or other traits vary across the landscape and vary through time. And so the evolution of these responses is often a fundamental means of local adaptation in natural populations. For a concrete example of this, consider two populations at two different altitudes, one at low altitude and one at high altitude, but at the same latitude. As you can see on the same calendar date, they're wildly different times of the growth season. And that's because the annual cycles of temperature and precipitation vary dramatically between the top of the mountain and the bottom of the mountain. But because they're at the same latitude, their annual cycle of photoperiod is the same. And day length is a critical cue that determines that many plants use to determine when during the year to flower, because it's a really reliable predictor of calendar date. The day length that is here in Rochester today is the same as it was on July 23rd 100 years ago, same as it will be 100 years in the future. But if the plants at high altitude were able to flower under the same photoperiods as the ones at low altitude, well, they'd be trying to flower through several feet of snow. Probably not the best idea. Likewise, if the plants at low altitude waited till the photoperiod, the day lengths that were permissive for flowering at high altitude, they wouldn't be able to reproduce because they'd be killed off by summer drought. And so natural selection has occurred to shape how these plants respond in flowering to the environment that they experience and locally adapt. And if we consider how these populations may evolve through time, well, with climate change in 50 years, the high elevation population may look like the low elevation population on the same calendar date, and it may be even drier and earlier in terms of its season in the future. And so by understanding the genetics of how populations have adapted to cope with variation in growth season timing across space, we can begin to understand how populations may do so through time as the climate warms. And so overall then, in terms of the questions we're interested in my lab, we're trying to understand what are the mechanisms by which organisms sense and integrate diverse environmental signals so that they produce environment-appropriate phenotypes. How is that coordination between internal and external rhythms adaptive and function ecologically to locally adapt populations? And then when we see these responses evolving, why and how do they vary? So we can look at questions like how many and what types of genes and mutations are involved in the evolution of plasticity to the predictable environment. And answering those questions gives us information about the ease and pace with which populations may be able to adapt to cope with a changing environment. We can also ask, when we see the same types of changes in plasticity evolve multiple times independently, either within species or between species, does this involve the same underlying genes? In other words, is evolution to the predictable environment driven by a predictable underlying genome? And then finally, it's also important to answer these questions because with climate change, some cues are going to be changing, whereas other cycling cues are not. And so we'll get novel combinations at different times of year or different times of multi-year time scales that are non-analog, that populations haven't seen before, and developing models will allow us to address how the populations may adapt into the future and also identify variants that may be important for that adaptation. Okay, so today I'm going to tell you two stories about how we've been studying the mechanisms and diversity of evolution of responses to the predictable environment. First, I'm going to tell you about a diurnal trait, turn, turn, turn. We'll look at the development, diversity, and ecology of solar tracking in sunflowers. And then the second part is to everything there is a season, looking at the evolution of photoperiodic flowering along elevation gradients in the common monkey flower. All right. So solar tracking is a really fascinating phenotype for many reasons, but one of which is the mechanism by which all of this is happening. So all of this bending is being driven by irreversible stem growth asymmetrically at different times of day on opposite sides of the stem. So if you follow stem growth using markers along the sunflower stem on the east and the west side, what you find is the east side of the stem grows faster during the day than it does at night. The west side of the stem is the opposite. It grows faster during the night than during the day, and thus you have differential growth rates that lead to that westward bending during the day and the eastward reorientation at night. In addition to this spatial and temporal complexity in growth, there's also developmental complexity, because sunflowers track from when they're young seedlings until they bloom, but as they approach anthesis, they move less and less west every day, such that by the time the flowers open, they achieve a fixed eastward orientation. And that's why when you see a picture of a blooming sunflower field, it looks like all the plants have just decided to pose for the camera. It's because they're fixed facing that direction. This actually led to some confusion early in the literature. When Europeans first encountered sunflowers through to about the turn of the 20th century, there was a debate about whether sunflowers were misnamed and that they thought they didn't track the sun. It's just because they were looking at the wrong developmental stage. And it was only when there was photographic evidence provided by Schaffner in the early 20th century that showed that yes, they track, it's just before they start to bloom. Cultivated and wild plants do it. And interesting, in the branched wild plants, the branches do it as well. And so this is a trait we've known about for centuries. It's what sunflower is named for. In Spanish and French, girasol and tournesol mean to turn with the sun. And you know, it's oft used as a metaphor in poetry. I will follow you like the sunflowers follow the sun. Even so, there's been very little study of the trait, actually. So between Schaffner and now, there's been few seminal studies, one of which I'll share with you, done by a Japanese group in the 50s, where they did a really pivotal experiment, pun intended. They took a plant in a pot, grew it up, so let it so it's facing west at dusk and east at dawn, and then rotated 100 degrees. Essentially, gave it the same jet lag as if I took you and teleported you to Mongolia right now. And then looked to see whether it could immediately sense its environment, reorient, or whether it was stuck on the trajectory it was originally following. And what they found is that it was stuck for a while. Instead of going facing east at dawn and west at dusk, it ended up facing west at dawn and east at dusk for quite a few days. And so they suggested that maybe there's some internal habit involved in this phenotype. But beyond this, there's been relatively little study, and so several years ago, Stacy Harmer from UC Davis and I decided to collaborate, and funded by NSF Plant Genome, we've been asking we've been trying to address many open questions about this interesting trait. So first, what are the cues that drive solar tracking and the proximate mechanisms that serve to integrate to result in this cool pattern of spatial and temporal complexity? How variable is solar tracking within and between species, and can we use that to get at the underlying mechanisms? And then finally, there have been lots of speculation out there about why they track and why they end up facing east, but these hypotheses haven't been rigorously tested, and so we've set out to do that as well. All right. So in terms of the cues that drive solar tracking, it's clear that a moving directional light source is necessary. If you grow sunflowers in the greenhouse, they grow straight up when they're top lit. And it's not only necessary, but Hege Batamian, a postdoc in the Harmer lab, has shown that it's also sufficient. And he did so in a cool growth chamber setup where he set up an arc of lights that would turn on sequentially to mimic the movement of the sun. And in this setup where they're just getting that input of the movement of a light source, they do recapitulate the tracking movement from east, the putative east lamp that turns on first to the putative west lamp that turns on at the end of the day over time. Okay, so daytime tracking, it's phototropic. Nighttime tracking, there's no equivalent light source that consistently tracks across the sky from west to east at night, so what's driving that? Stacy and I hypothesized that perhaps it's the circadian clock, this internal oscillator that all organisms have that orchestrates or gates many activities so that they occur at particular times of day or night, particularly when they have to mobilize resources or involve developmental processes in anticipation of a particular time. And so we hypothesized that that could be involved here. And so Hege did a couple additional experiments to implicate the clock. First, he did a classic experiment where you take plants in a cycling light environment, and then move them into constant conditions where they don't have those external cycles anymore. And when he moves the plants into constant light, the solar tracking oscillation continues, though it damps over time. The other experiment that he did was able to take advantage of the fact that in that arc of light growth chamber, not only is the daytime tracking recapitulated, but when the total day-night cycle is 24 hours, they're also moved back from the west lamp to the east lamp during the night. However, when he experimentally manipulated the day-night cycle to be longer than 24 hours, to make it 32 hours, the plants still tracked from the east lamp to the west lamp. But at night, they stopped moving back from west to east. They only moved back in the morning. When the morning lamp turned on, they realized, oh, I really ought to be over there. And interestingly, if you follow those same plants, but change the growth chamber settings back to 24 hours, the nocturnal reorientation resumes. And we see the same thing happen when we transfer plants to a shorter 16-hour day length and then move them back to a 24-hour day length. And so because there's this 24-hour sweet spot, that really implicates the circadian clock as being involved in this nocturnal reorientation. And so Stacy and I really think this is a wonderful model to begin understanding how internal rhythms from the clock and external rhythms and light signaling drive complex patterns of plant growth in a dynamic natural environment. And so the question then becomes, all right, if these are the cues, how are they integrated to give rise to these interesting patterns of stem growth. And my lab has been taking a natural variation approach to this problem where we use the diversity out there in cultivated and wild sunflower to genetically map variants that affect this trait. We've done so using two mapping populations, one cultivated and one wild. The cultivated population is an association mapping panel of about 280 cultivars developed by the International Sunflower Genomics Consortium. This panel is fully resequenced, so we know throughout the genome where the genetic polymorphisms are, and so any phenotype that we measure on this panel, we can then immediately associate with SNPs and ask what are the underlying genes associated with variation in our phenotype. So we've done this for solar tracking. We phenotyped the panel for solar tracking. We grew plants in the field in these 5-gallon paint buckets, doing a product placement experiment for Lowe's that I'm still waiting for the check for. We then moved them to a backdrop where we filmed them with hunting cameras for 5 weeks to develop the videos like the one I've been showing you. We then score these videos for traits like when the plants start moving toward west relative to dawn, or when they start moving back toward east relative to dusk. And we also scored the position that plants face when they bloom. And of course, because we're doing time-lapse imaging, we capture a lot of other interesting phenotypes as well. So to give you a sense of the variation we see here, thinking about when plants start moving back from west to east relative to dusk, you can see that across our 280 lines, there's a nice normal distribution centered around dusk. But there's variability here, and if we split this up among all of the individual lines, what you can see is that yes, many of them fall around dusk, but there are others that start moving back over an hour earlier than dusk reliably, and others that start moving back over an hour later than dusk reliably. In terms of the final head orientation, yes, in both Charlottesville, where we grew the plants originally, and then in Iowa, where Laura Merrick, the USDA oilseed curator, was generous enough to phenotype this for us as she was bulking the population as well. On average, they end up facing east, but there's variation around that mean among the lines. And notably, these distributions are not really correlated between the populations, between the locations, suggesting that there's strong G by E for this trait. Okay, so we've got our phenotypes from the sequencing data, we have our genotypes, we can do association mapping. And so my postdoc, Serenity Hololu, has been pursuing this. Here are some Manhattan plots showing for all the SNPs that we include across the 17 sunflower chromosomes, the strength of their association with phenotype. What I want you to take away from this are 3 things. Number 1, there are peaks. That might not sound exciting to you, it might sound trivial, but it's really exciting to me because when we went into this, we had no idea that there would be variants of large enough effect at high enough frequency in the population that we'd actually be able to detect anything. So that we see something, it's just like, oh great, this is good. Number 2, the peaks are really narrow, so they span maybe 1 to 5 genes at most, so we have really nice precision to identify candidate genes in these intervals. But 3, I'm not going to tell you what those candidate genes are because there are some interesting things that might have novel functions to annotate here. There's no screaming clock or light or hormone gene to go after. Where we do have those candidates is from the second panel we grew, which is a cross between 2 wild individuals, one from Manitoba and one from Kansas, that we took out to the F4 generation so we could incorporate as much recombination as possible. We grew them in the field, still waiting for that check, and phenotype them using the same methods. These were not resequenced already, so we used genotyping by sequencing to genotype these plants efficiently ourselves. This was done by my postdoc Karen Barnard-Cabot, and when she puts the genotype and phenotype data together, again, we do quantitative trait locus or QTL mapping, and we find regions of the genome associated with traits such as when they start moving toward west or start moving toward east relative to dawn and dusk respectively. And these windows are wider. There may be 50 to 300 genes under there because there's less historical recombination built into the population. But under these regions, there are quite a few really exciting candidate genes whose homologs in Arabidopsis are involved in phototropism, auxin signaling, and transport, as well as stem growth. And so to follow these up, what we did last summer in the field was we wanted to look at gene expression of these genes in the parents of this cross. And so we took advantage of a tool that I think every person should have, which is a vegetable peeler. And we peeled the east and west sides of the stem's epidermis off, and then isolated RNA from that to do qRT-PCR of our candidates. And so here are the results from that work. What you need to pay attention to is when lines of the same color, so from the same side of the stem, differ between the Kansan and the Manitoban parent in solid or dashed. And what you can see for these candidates are some interesting patterns. So for instance, this IVH-like gene shows a difference specifically at dawn on the east side of the stem between the two parents. And this maps to a QTL that affects the timing with which they start tracking toward east relative to dawn. Again, here with this pre, we have a difference specifically on the west side of the stem between the two populations at dusk. And then we have a couple genes that show differences at most times of day between the two populations. And interestingly, these two genes are both in Arabidopsis involved in a complex that sits right on top of whether cell elongation is deployed. So they act as a switch point depending on how they interact with each other, suggesting that right where these cues, and that switch point involves the interaction of light and auxin signaling. And so that suggests that this switch point is really perhaps what varies among population and is getting us to the mechanism by which this integration is happening. And notably, this does sit downstream of what's happening with the clock, because if you look at key clock components in both populations on both sides of the stem, there's very little difference in expression between them. Okay, so that's as far as we've been able to get in terms of getting at the underlying mechanisms driving tracking. Now I want to turn to the ecological function of tracking and why the plants face east. And we've been able to make some progress on this first question and more progress on the second question. In terms of whether tracking provides a benefit, we believe we've shown that it does. And we're able to do so because when you stop plants from tracking, they put on less biomass and less leaf area than control plants. And we did this both by tethering plants to stakes so they could not track. Sometimes they wanted to track so much that they would actually snap their own stems rather than stay up straight. Hagop also did an experiment where he did what the Japanese researchers did and turned them 180 degrees at dusk, but he didn't just do it once. He tortured them and gave them jet lag every day until they stood straight up. It's like the opposite of when someone spins you for the piñata. Rather than falling over, they just get all rigid. Anyway, either case, plants that don't track put on less biomass and less leaf area. But the question is why? How does tracking help? We don't really know. We're trying to pursue it further. But one small insight we have is when we look, could come from diversity. So last summer we looked at the diversity of tracking phenotypes across the Helianthus genus. It turns out that solar tracking appears to be a synapomorphy of the genus. It's been lost a couple times, but there's no real phylogenetic signal there. But when we look within Helianthus annuus, what we find is that there's a nice trend that the timing with which plants start moving from east to west relative to dawn shows a good correlation with the summer precipitation at the site of origin. And so what I think this might be indicating is that there's a tie-in between the diurnal regulation of water relations that are determining plant water potential and how plant water is moving through a plant to drive stem growth and the timing with which they're tracking. Okay. We've made more progress in terms of thinking about, learning about why plants end up facing east when they stop tracking. And this was started as an undergraduate thesis by my first undergraduate, Evan Brown, and followed up by a technician, Austin Gardner, another undergraduate, Lane Scher, and Nikki Crew, a postdoc in the Harmer Lab, who replicated the work in California. And what Evan did was really simple. He grow plants up till they bloom, leave some facing east, turn others to face west, measure a bunch of things and see what differs. Okay? The first thing that differs is probably not so surprising. East-facing plants warm up quicker in the morning than west-facing plants. And we can see that both with forward-looking infrared photography as well as with data loggers hooked up to thermocouples. Okay? But in terms of how this affects any aspect of plant performance, we measured lots of things. And the only thing that was consistently different between populations across trials, across multiple years, and between Virginia and California, was that east-facing plants attract greater pollinator visitation, specifically in the morning, than west-facing plants. And this was an interesting result that raised several questions. The first was, is this difference in temperature responsible for the difference in pollinator visitation? So we did the experiment again, but we added additional treatment where we used radiant heat lamps like you might sit under on a patio at a restaurant that were tuned by a propane tank to warm up west-facing plants to match the temperature of the east-facing plants. And as you can see, we're able to match that quite nicely. And when we look at pollinator visitation, when we heat up the west-facing plants, they recover a good deal of the gap in pollinator visitation between west and east. The second question that this raised for us is when we grew the plants in Virginia and measured seed set between east and west-facing plants, there was no difference. So does this really impact fitness if there's no difference in seed set? Well, seed set's just a measure of female fitness, and we speculated that because this involves pollinators, maybe male fitness is being affected. And so we did an experiment to see whether eastward orientation impacts siring success. And we did so taking advantage of a genetic tool in sunflower, a cytoplasmic male sterile line, a line that cannot produce its own pollen and needs to get fertilized by another plant if it's going to set seed. We put these plants in the center of a ring of other plants of two genotypes we could tell apart by molecular markers. We faced one of those genotypes east, we faced the other west, unbagged the sterile plants, let the pollinators go at it, collected the seed, grew up the seedlings, got the DNA, and then did the Jerry Springer Who's Your Sire test to ask whether the east-facing plants sired more progeny than the west-facing plants. And indeed, in 6 of our 7 trials, 5 of them significantly, east-facing plants did sire more offspring than the west-facing genotypes, indicating that orientation does impact male fitness in sunflower. Okay. So overall then, in terms of the genetics and ecology of tracking, I've shown you that directional light drives the westward movement during the day, and the nocturnal reorientation is dependent on signaling from the circadian clock. Natural variation exists within cultivated and wild sunflower that's allowing us to identify cool candidate genes involved in tracking, and that both tracking and its cessation enhance sunflower growth and reproduction. Okay. So that's all I'm going to say about this diurnal rhythm, and now I'm going to turn to another way that plants respond to the predictable environment and how those responses evolve. This in terms of photoperiodic flowering and seasonal plasticity in the common monkey flower. Before I get into the specific system I'm using here, I want to take a little step back and address another facet of how organisms respond to the environment. So many plastic traits are actually threshold traits, and that the phenotype is only expressed when organisms when the environment is above some minimum threshold or falls below some maximum threshold. And we often think about threshold traits in the context of polyphenism, where there's 2 alternative phenotypes, either of which is expressed depending on the environment experienced at a previous developmental stage. However, threshold traits also play a key role in life history transitions, because whether it's going from seed to seedling, or egg to hatchling, or rosette to flowering, these transitions are often obligately dependent on receiving the correct environmental information to proceed. Now, in terms of understanding how threshold traits evolve, this type of quantitative trait has received less study than many other types of quantitative traits. So basic questions such as how many and what types of genes are involved in the evolution of threshold traits, we don't know the answer. So do they change by few genes of large effect or many genes of small effect? Likewise, when we see the same shifts in threshold happening multiple times independently, is this a result of similar changes at the underlying genetic level? And this question about convergent genetic evolution has been asked many times in the context of lots of traits, but it takes on a special significance in the context of phenotypic plasticity. Because one of the early models for how plasticity should evolve, the allelic sensitivity hypothesis, posed that you should see repeated substitution of alleles of different sensitivity to the environment at the same locus repeatedly, sort of tuning the response like a rheostat. So we should see the same, under that, we should see the same loci involved over and over again. The final question about the genetic architecture of evolution of threshold traits that comes up here is whether the threshold that determines whether or not the trait is expressed will necessarily evolve in lockstep with the value of the trait when it is expressed. To understand what I mean here, first consider a continuous reaction norm in a coarse-grained environment. So if plants are experiencing selection to reduce the value of the trait in this particular environment, then by necessity, it's also going to change the parameters of the reaction norm, in this case, the slope. However, with the threshold trait, there's no reason a priori to expect that when selection is acting to reduce the value of the trait when it is expressed on the y-axis, it's also necessarily going to change the threshold that determines whether or not it's expressed on the x-axis, unless those 2 things are controlled by the same genetic variation variant phytopically or variants controlling both are tightly linked. So there are genetic correlations that could lead to the correlated evolution of both of these parameters. And this is more than just some esoteric question about how quantitative traits evolve. It has real ecological impact. Because if these two parameters can evolve independently, then are controlled by different variation, then natural selection is free to locally adapt these responses to any individual environment. There's no constraint. In contrast, if they are genetically correlated, while that might cause some constraint, if selection is acting to maintain variation in the trait value within a population, then it's also maintaining variation in the threshold within that population. And so if populations colonize new environments, or if the environment changes, they have that segregating standing variation ready to go and respond to that change. All right. So to address these questions about the genetic architecture of threshold trait evolution, I've been working in annual populations of the rockstar model system, formerly known as Mimulus cutatus. It has an obligate long day response. Plants only flower when the day length exceeds some critical minimum. Below that, they stay as little cabbages forever and ever. Above that, they will flower quite rapidly. And previous work in the lab had suggested that that threshold varies among populations, and perhaps there was a geographic trend in that. And so several years ago, I went on a wonderful road trip where I started down in San Diego and went up to Eugene, collecting elevation transects of populations up and down the Sierra Nevada and the Cascades mountain ranges. I collected these plants, brought them back, grew up one generation in the greenhouse, and then grew up another generation under a battery of photoperiods from very short to very long to measure the critical photoperiods of these populations. I also grew them up under the greenhouse under inductive conditions where everybody flowers to measure time to flower in inductive conditions. Okay, so here's what I found. Indeed, there is a nice geographic trend in that high elevation populations require longer day lengths to flower than low elevation populations. And here elevation is really a proxy for climate, and that it's cooler and wetter at high elevation, shifting the seasonal timing later in the year. Okay, so we have variation among populations. It's distributed in interesting and adaptive ways. What's the genetic basis of this variation? Again, to approach this, I took a genetic mapping approach where I crossed multiple high elevation populations by low elevation populations, creating a series of F2 mapping populations that I then grew in growth chambers under conditions where the high elevation parents would not flower, but the low elevation parents would. When we look at the distribution of flowering in the F2s, we see that some F2s flower, but some do not, and because there's segregating variation, we can map it. Again, we used a reduced representation approach, in this case multiplex shotgun genotyping, to efficiently genotype many individuals at hundreds of or thousands of the same SNPs across anyway, we did this for 4 populations, and they fall into 2 nice pairs. So 2 from the Cascades and 2 from the Sierra. And in these pairs, both the high elevation parents have the same threshold and both the low elevation parents have the same threshold, allowing us to address this question of convergence at the genetic level. All right, I'm going to show you the QTL mapping results just for the Cascades for the sake of time. Here we have the South Cascades and North Cascades crosses, all the markers we were able to genotype across the 14 chromosomes in the genome, and the strength of the association between whether or not the plants flowered and that marker. And what you can see is that in each of these mapping populations, there's just 3 significant QTLs, one of which is shared between the 2 populations. And we think that this is likely most of the QTLs, if not all of them, because if you add up the number of low elevation alleles that a plant has in this population, so 3 loci, diploids, 6 possible alleles. If you go from all all high alleles to all low alleles, you go from 0% flowering to 100% flowering in both cases. Okay, so we found equivalent numbers of loci in the 2 Sierra Nevada crosses that we looked at. And when we look across all of these panels, that one shared QTL was the only one shared within a region, and it was really the only one shared between any 2 crosses. So this indicates that there's lots of different loci, at least 9 different loci, that can independently jigger whether or not the plants flower under a given photoperiod And there's little evidence for an allelic series where you're repeatedly substituting different alleles at the same locus over and over again. Okay, finally, to address this question about whether there are genetic correlations caused by pleiotropy or linkage that would cause trait values and their thresholds to evolve in lockstep, we were able to look at this by growing up full sieves of these mapping populations in the greenhouse where everybody is induced to flower, and ask whether the regions of the genome that explain variation in days to flower under inductive conditions overlap or not with the ones that affect photoperiod threshold. We did this for 2 crosses, one from the Cascades and one from the Sierra, and here's the results from the Cascades. So what you can see in purple are the QTLs I already showed you, the regions of the genome that affected whether or not they flowered under 13-hour conditions. And in black are the QTLs that impact the time to flower in the greenhouse where everybody is flowering. And as you can see, these QTLs don't overlap. They're on entirely different chromosomes. So there's a unique genetic basis for variation in both traits. And that was true in both this cross and in the cross from the Sierra. And so this really suggests that these 2 aspects of seasonal flowering evolve through using independent variants. And it suggests that there's lots of variation out there that local adaptation can use to fine-tune the reaction norm to the local environment. And I think that's not just potentially what could happen, but it's what has happened and has been important for the expansion of this species across its range. Because unlike critical photoperiod, which I showed you shows a monotonically increasing value with both elevation and with latitude, the relationship between flowering time in the greenhouse, so there's a changing relationship with elevation depending on latitude. So in the Sierras, high elevation populations take longer to flower than low elevation populations, but it's the opposite in the Cascades. High elevation populations flower earlier than plants from low elevation populations. And what I think is going on here is this is all driven by a really complex set of seasonal growing seasons across this range. So at low elevation in California, like in the Central Valley, the season is bounded by drought on both sides. Plants germinate in the late winter, early spring rains, and they have a short period to flower and set seed before the onset again of summer droughts. As you go up in elevation in the Sierra, or as you go north to Oregon, now snowpack downs the beginning of the growth season, but plants have really long, lush, damp springs to get big and produce more flowers before the onset of summer drought. Whereas as you go all the way up to the mountains of Oregon, now that snowpack sticks around for a really long time. Plants only start germinating in late May, early June, and have maybe 6 to 8 weeks before they're toast due to summer drought. And so while the start of the growing season moves later and later in the calendar year as you go up in latitude or elevation, and that's tracked by critical photoperiod, the duration of the growing season goes from short to long to short again, and that's what's being tracked by time to flower in the greenhouse. And so the fact that these 2 things can be genetically dissociable likely has facilitated the adaptation of this species to its really broad geographic range. All right. So overall then, I've shown you that there's lots of variation in critical photoperiod and flowering time among these populations structured along elevation gradients. There's few loci that are responsible for the divergence within any given transect, but we see really few, we only saw 1 locus that was reused multiple times across this range. And I think that's really interesting, because for many other traits we've seen repeated use of the same genes to change phenotype independently through natural selection. And it raises 2 possibilities for me for why we're seeing this. The first is that there could just be really great developmental flexibility in the regulation of this trait. You tweak something here in the network, you tweak something there in the regulatory network, you get the same evolutionary and phenotypic counterpoint. The other possibility, and it's not mutually exclusive, is that that local adaptation is really local, and only by getting the particular combination of allelic variation in that population do you hit upon the optimal critical photoperiod for that habitat. The implications of those 2 possibilities are quite different. So if there's lots of developmental flexibility, lots you can do, there's adaptation to climate change should be relatively easy. Whereas if it's strong selective constraint and you have to hit on particular combinations of alleles, then adaptation to climate change may be much more difficult. And so to address these questions, I am now involved in a collaboration with Dan Runcie at Davis, Jay Sexton at Merced, and Akiko Satake at Kyushu University, funded by NSF to study this question, understand how this is happening. First, by identifying the underlying allelic variation. Second, by doing transcriptomics to understand how these individual QTLs shift thresholds. And then finally, bringing these plants out to nature and understand, do these regions that affect flowering under particular photoperiods in the growth chamber also affect flowering and fitness in field environments. So on the first question, we've made some progress, and this has been pushed by my postdoc, Nick Kuyers. And again, like for solar tracking, we have some candidate genes underlying our QTL regions that we've been going after to characterize. But the thing with candidate genes is usually you're like, yes, this is great, but sometimes you get more than you bargained for. We've been particularly following up on this one here, where there's there's a cluster of homologs of flowering locus C and MATS affecting flowering genes, which are major repressors of flowering time. It's not just that there's a cluster, but there are 11 closely related homologs of these genes in this 1 megabase pair interval. So there's lots to dissect, and it's really hard. But Nick is bashing his head against this for a while, and he's been able to show the following by looking at gene expression of these paralogs under conditions where the low elevation will not flower, but the high elevation parent will. And what he's found is that when we look at the parents of the Southern Cascades cross, several of these paralogs show higher expression in the high elevation parent than the low elevation parent. And this makes sense because this is a repressor, so it should be up higher in the plants that are not flowering. Okay? When we look at them in the Northern Cascades cross that also show this QTL, what we see is, yes, some are upregulated, but really all of them are upregulated. So even though this is a shared QTL, the underlying genetic mechanism may actually be quite different yet. And we want to get into this further, and for a while I was despairing about our abilities to actually dissect what's going on here. We've done some work to show that there's cis-regulatory variation affecting this, but really we need functional genetic tools, and I'm happy to say that my postdoc, Serenity Hololu, has made great progress in this. We've been able to now knock down gene expression of genes involved in pigmentation, here patterning of the nectar guides using hairpin RNase, RNA interference. We can also look at allelic differences in promoter activity using reporter genes. And then the key thing for dissecting all of this is that we now have CRISPR-Cas9 gene editing working in sunflower, shown here, or sorry, in monkey flower, not in sunflower yet, where we again have been able to recapitulate this phenotype that alters the expansion of petal spots in the nectar guides. So we're hoping to use this to get more of this further. Finally, turning to this question about whether the genes involved in critical photoperiod regulation in the growth chamber affect flowering and fitness in the field, Nick has done field experiments at low and high elevation in Oregon, where he's grown up plants that we've bred to be homozygous for the low elevation allele or the high elevation allele, or both, at 2 of the loci involved in the cross from this region. And he's grown them up and asked whether these genotypes influence fitness in the field, and what he finds is, at least in this one field season at low elevation, one of the loci does, the low elevation allele does increase survival to flowering and flower production at low elevation, suggesting that this region of the genome is critically important for fitness as well as for flowering time regulation by photoperiod. Okay. All right, so then overall, thinking about the evolution of plant responses to the predictable environment, what I've shown you for solar tracking is that the interaction of internal rhythms with external directional light signaling enhances both plant growth and plant reproduction in sunflower. And then I've shown you that there are diverse genetic changes that alter really individualizable aspects of how plants flower in a seasonal manner as populations have adapted to a really broad range of complex geographic variation in climate in California and Oregon. And so hopefully this has given you a good understanding of how we in my lab approach natural diversity and understanding, really from the nucleotide level all the way through fitness, really starting at phenotype and working out in both directions. how the evolutionary process and the evolution plasticity happens. We also have some great projects on domestication that I haven't had a chance to talk about. But what I want to emphasize in closing is that this work is really integrative, and I've really benefited and been able to do this through my training from many mentors who like taking all of these approaches. It really started with my undergraduate honors thesis mentor, Ward Watt, who is always about looking at the recursive helix of evolution, understanding how genes affect protein design, affect plant, organism performance, affect fitness, to then change allele frequencies. Over time, I've also benefited from the fact that I've been co-advised by multiple mentors, whether it was as an undergraduate, as a graduate student, or as a postdoc, who helped me bring both molecular and mechanistic perspectives, as well as diversity and evolutionary perspectives to the work that I was pursuing. And of course, we don't get everything that we know or develop into the scientists we are because of our mentors, though they play a large part. I've also benefited from many wonderful labs that my mentors have been able to recruit and cultivate, and I have learned greatly from all of these people over time, many of whom are not even shown here. And I've tried to replicate this in my own lab, and much of the work I've shown you is, of course, driven by the people in my lab rather than myself. And so, oops, there we go. No, there. And so I've tried to highlight their contributions as I've gone along. I'm highlighting particularly people who contributed, but there are many great people in the lab. And I especially want to thank all of the many undergraduates who spent so many summer hours in the heat hauling those buckets back and forth. Super happy. And then finally, in all of these projects, I have wonderful collaborators who I am so happy that they are willing to collaborate with me on these things, and I wouldn't be able to do this without them, and thank the funding sources who have supported the work as well. And with that, I have to plug the Citizen Science Initiative that we've started as part of the broader impacts of our NSF grant on monkey flowers. We are working with the National Phenology Network, particularly this is headed up by Kathy Gerst and then Nick Coyers, piloting a phenological monitoring program along the Pacific Crest Trail, particularly along the segments in Yosemite and North Cascades National Park. So if you know anyone going later this summer or next year, send them to this website. And with that, I'll let you bask in the monkey flowers and take any questions. Thanks. Yeah, Stacy.
So it seemed like in both of the sections of your talk that there were different genetic bases for the traits, like across and then across the accessions and then again. And I wondered, even though there are different loci, do you think there are going to be themes? Are these going to be mostly transcription factors? Are these big? My case, big transcription factor families.
Yeah.
Is regulatory changes at those?
So I think that, one of the themes, at least for the monkey flower work, is there's going to be a bunch of copy number variation. So I highlighted that to some extent with the flowering locus C, and that's affecting flowering genes. But several of those other QTLs are either partial or full homologs of flowering locus T genes or things in that family. And so I think that there's some aspect of how gene duplicate copies are coming on or off, or all of that that's feeding into the system. So I think that's one major trend. In the solar tracking work, I think it's really cool that we've been able to find things that likely function in the switch point that integrate multiple signals and then target downstream cell elongation, though we need to suss that out further. And I'll be interested to see what the novel things that we're finding from the cultivated work are. But yeah, part of me thinks some of this is due to really these sort of, as David Stern, who I took my title from and edited a little bit, calls them switch point genes or hotspot genes. I think some of that's going to play a role, but I think there's also a lot of other tweaking that's going on, particularly for the photoperiod stuff. Yeah, Chelsea.
I I wanted to know, I have two questions.
Yeah.
The first one is, since you haven't heard from Lowe's, have you considered switching to Orange Home Depot buttons?
They are closer to my new field site, so probably. Yeah.
I was actually wondering if the color had anything to do with the soil temperature because they're buttons. But my real question was, so you did the epidermal peels and the sunflower tracking.
Yeah.
Do you see any changes in the internal anatomy in the vasculature that could also be interesting for?
Yeah, so we haven't looked specifically at that. One of the reasons we went to doing the peels is that Hagop, when he was doing, just on one line, doing RNA-seq studies to see what differed between East and West, he did it once, doing all of these extractions, just taking half of the stem, the East half and the West half. And he got really muddled results, and only like a couple things were actually differentially expressed, which is like the opposite of what you typically see in RNA-seq studies, where usually you have a plethora of too many things competing for your attention. And so then they repeated it just using the epidermis to isolate particular cell types, and that seemed to give much nicer results. We haven't looked at the internal anatomy. Hagop did try at least to look at the epidermis and look at whether this was being driven, really to show that this is being driven by cell expansion, which is what I expect is going on, versus cell division. But that work is still in progress. Yeah. Yeah.
You mentioned that there's been losses with the hemianthus tracking.
Yes.
Can you talk about that?
There's been a couple losses, mainly in lineages that spend much more time as a rosette and then bolt really quickly. And so, you know, they just sort of invest in growth. It's not universally true, but that's one pattern in the data. Yeah.
When you did the false heating of organization, you didn't get a full recovery of the visits. Do you know what else? Or do you speculate what else?
I'm not sure what else necessarily is going on, but one of the other complexities here is I think there's more going on than just the heat reward. So one of the things that Nikki has noticed is that when you turn the sunflowers to face west, the timing with which the florets open and present their pollen in the morning, or those new stigmas come out, is delayed. And so there could also be, this also could be because there's a delay in pollen reward. And so, you know, if we think about solar tracking as a paradigm for understanding how internal rhythms and external factors that are cycling are integrated to regulate stem growth, thinking about floret maturation in the disc is another paradigm where we can think about how the circadian clock is involved, because when you move flowering plants into constant dark, they still keep cycling with their flowers opening at the same times of day. But then when you change them from east to west, you get this delay. So clearly some environmental input is involved. And so we've been recently recommended for funding to follow this up by studying the evolution and mechanisms by which the circadian clock and temperature and other environmental factors influence the timing of floret maturation and how that impacts pollination and cell-phane.
So let's think it's 11 o'clock. So